We'll move on to our next presentation from Bradley Alicea, who is at the Orthogonal Research and Education Lab as a head scientist. And he's also a senior contributor to the Open Worm Foundation. You can find his Google Scholar in the chat box. And he'll be discussing how you can incorporate the beauty of development into some artificial neural networks.
Yes, so thank you. Jesse Parent, who's a co-author on this, is also in the chat. So if you want to ask him questions after, he might be able to answer some. Thank you to our co-authors listed here. And this is approaching artificial intelligence as an embodied developmental process. So these three GIFs here show different organisms in the process of development. Here we have some neural crest migration. Here we have a developing nervous system in the axolotl, and there we can see elongation of the zebrafish notochord. So we notice that all these things are very much morphological or involve the body in their development. And so we're going to take a little bit different approach than what we would have with biological organisms. We're going to use something called Breitenberg Vehicles. So if you're familiar with this, this is a toy model that was developed in the 1980s by Valentino Breitenberg. Their first order stimulus response mapping. So you have this light source up at the top. The sensors are in the front of the robot. They get transduced to the wheels, and it drives it's called a vehicle, but it's really implemented usually in robots, and it drives it in different directions, and they exhibit all sorts of behaviors. So they yield these sort of tactic or reflexive behaviors. And so it's based on a very simple neural network. But we've taken this a step further in our group. We've developed something called developmental Breitenberg vehicles. So these are embodied nervous systems in these vehicles, and the neural network develops from that simple sensor effector mapping to something more complex within the organism. So this is what we call network morphogenesis. So we're building a network from nothing, and the initial conditioning is that simple mapping, but then you get a complex network over time that then behaves in various ways. So this is a schematic of how this works. So we've used genetic algorithms to develop this network morphogenesis technique, where we have layers of cells, which are like neurons. These serve as interneurons. So the sensor is up here, the effector is down here, and these circles are interneurons that are layered, and then they connect gradually over time. They connect in different combinations of pathways. And we can add other types of models, such as a CGS, which is something that we use in our group as well. This is a more mature version of this developmental Breitenberg vehicle with a denser set of connections and maybe a couple more internal nodes or interneurons. And so we can do different types of behaviors with these. One example I use here, used it in a number of talks, is called Chromotaxis. So the idea is these vehicles will be attracted to not only a certain color, but can differentiate between colors. And so as a phototaxis, this operation is simple attraction or repulsion. Are you attracted to this color, or are you repulsed by it? As a preference, you have this separate continuum of emotional valence. So now it becomes important to differentiate between different shades of a color or different colors. And then there's a difference between congruent stimuli. So color, for example, and then maybe the shape of the color might become important. And we've simulated that using generalized Hebbian learning, which occurs later in development. And then we can also have differentiation between incongruous colors, and this enables spatial learning. And this is something we've done with a genetic algorithm, not only evolving these networks, but like differentiating between things in the environment as well. And actually, I'll send you this paper. This is a good source for this type of work. So the heart of this method is something we call connectivity activation encoding. And this is a simple notation for a matrix. And this is what it looks like. You have your cells and you have connections, and that's your matrix. Each developmental time point produces a connectivity matrix. But of course, as the size of the network increase, or the number of interneurons increases, the size of that network also increases. But it's it's basically over developmental time, you keep adding neurons and adding connections, and that's what you end up with. And then you have, from that matrix, you can get a corresponding determinant A. So then we have this relationship between the matrix growing over time and these different A's that are available to over developmental time. So in development, the number of neurons can grow and synapses can fluctuate in terms of their strength and efficacy. We think of this in terms of universal functions rather than as an actual transformation of Wij. It's this idea that they're like these fundamental rules that govern these matrices no matter what their size. And so that's borne out a little bit in the deep learning literature. But this is very different from sort of like a reductionist representation. In that case, you have all sorts of different things going on in the brain. What we need to do is to make this Wij sparse, but we also want to make it as biological as possible. We want to add biological sort of relevance to it. And so you can see how this kind of works in a C. elegans model. These dots, white dots, are the number of neurons that grow or that appear or born in the C. elegans over developmental time. I've mapped them onto an adult to show that you start with a single neuron in what will become the head, then you get a couple neurons in what will become the head, and then finally you get many neurons in the head, and then some starting to be born in what will become the tail. And that's how we envision our vehicles being very similar to that. But we also add something called critical periods, and critical periods are a window of developmental time that features an enhanced ability to learn. So critical periods exist in all sorts of organisms, and we usually associate it with like early visual learning or other types of learning and physiological reorganization, which is actually one of the hallmarks of critical periods. And so we are going in this direction where we're taking these vehicles, they grow over developmental time, but we're introducing these critical periods where the critical period can expand or contract, and that's when a majority of your growth and reorganization occurs. And so this is mapped out in the following way. You have morphogenesis, which is defined by slow growth in this network. So you add nodes and you add connections, but it's relatively slow. Then later on, you get this critical period, which is in the dark gray. And you can see the difference between this morphogenesis and the critical period, which is technically part of the morphogenesis, is where you get your number of nodes will grow at a higher rate, and you'll get a lot of reorganization amongst the connections between them. Then finally, you get developmental learning, where you stop adding nodes altogether, and then you actually modify the weights of the connections. You don't lose and gain connections so much, you just modify the weights. So in that kind of stable network, you can start to learn things. And so this is what we call developmental freedom, which is where you've fixed the network in terms of its morphology, but now you're learning on it and you're changing the weights with respect to like different sorts of stimuli. And so we can think of this as terms of early and late effects. So we have this contingency matrix here that we've made where we look at the critical period and then we look at developmental freedom. So an early critical period means that the adult neural network tends to be more homogeneous. There's less time for preliminary or auxiliary nervous systems or behavioral traits to develop. And there are fewer varieties of experience prior to maturity. Contrast this with a later critical period, which is what we see in the adult neural network in that case, is that it can exhibit a larger number of possible configurations across the population of individuals. Slower developmental periods afford more time for exploration and wandering. Then if we look at developmental freedom, we see that there's a low degree of overall developmental freedom when you have an early critical period, and then of course correspondingly early developmental freedom. And this early critical period allows for faster ascendance to a fully mature suite of behaviors and capabilities. With late developmental freedom, which corresponds to the late critical period, you have a high degree of overall developmental freedom, and this allows for a slower ascendance to adult behaviors and capabilities. And so I'm going to also introduce, before I wrap up, this idea of Gibsonian information. This is the way we view the way we view information coming into this embodied nervous system. And so the key to Gibsonian information is that motion provides information. So what that means is it could relate to optical flow or relative motion between the object and observer. And I'll give a couple examples. So this is basic visual flow with motion and contrast cues. You can see this roller coaster. If imagine the agent is on a roller coaster and it's experiencing all these motion cues. But you can also do things like look at a disjoint distribution, and that is between a moving stimulus and a stationary stimulus. And in this case, you have this white noise embedding where you have just a stable white, static white noise background versus a dynamic white noise background. And you can see their motion cues in the ladder up at the top. Then you also have time series structure. So you have things that happen, events that happen in sequence. And so when they happen out of sequence, or you cut things that are expected out of the sequence, you get different types of visual scenes and different types of responses in the nervous system. And then finally, maximum versus minimum coherent movement. So this one's a little tricky, but this up at the top is the random movement of these balls. And so this is minimum coherent movement because while there's a lot of movement, it's not coherent. Contrast this with at the bottom where you have a lot of maximum amount of coherent movement. You have these flocks of birds that are moving in a very patterned way, and you can see exactly how they're kind of coming together and clustering. And so these are all the things that we want to be able to train our agents on and get them done, get them to differentiate between these different types of information, and then ultimately make that quantitative. We're also working towards reinforcement learning approaches, which are where we can develop discrete layers that will help the agents learn better policies. The growth of these nervous systems have to be countered by a potentially large and continuous action space. And we're thinking in terms of more specifically curriculum learning, where you give easy examples and then you give harder examples over time. And then you end up with, an ability to learn more and more over time. Finally, there's this issue of contingency in development. So going back to the scenarios for developmental freedom in the critical period, we have to remember that development unfolds in a certain contingent way. So each node is a developmental event. And so when you put a node in a certain place, you constrain the development of future phenotypes and how they can learn. And so you have these pathways that are contingent upon previous steps. And so when we build a nervous system, we have to consider that contingency is important. So morphogenetic events actually restrict the network. The more morphogenetic events you have, the longer your morphogenesis period goes and the later your developmental freedom is, then you have this more contingent and more restricted network. The more changes will occur during the critical period, but it results in a network restricted to an increasingly smaller possible outcomes over time. And then finally, developmental freedom may allow for a limited convergent evolution of development. And this may have, there may be ways to investigate brain versus mind using that approach. Finally, thank you for your attention. Our work is sponsored by the Representational Brains and Phenotypes group, and we do this Saturday morning NeuroSim group every Saturday morning. We're open on the web here. We have a GitHub repo that if you want to contribute to or look over our work, we have that. We also presented something called the Psychophysics of Non-Neuronal Cognition earlier today, which might also be of interest if this talk was interesting to you. And you can read our preprint on this work on ResearchGate. Here's the QR code and the link. Thank you.
Thank you, Bradley. Nice talk. So we just have a couple minutes for questions. We have one here from Drobomir. These were really fun examples of Gibsonian information. Could you please say how you would investigate specific forms of information in your robots?
I think presenting, like when I gave the example of Chromotaxis, for example, that's where we're taking things from the Breitenberg vehicle and making it a little bit more complex, having multiple parts in the stimulus and then testing it in that sort of world. With Gibsonian information, we probably look at some of the things like, some of the examples that we used in there, like taking a sequence that it might learn like a sequence of events, and then chopping up the events, mixing them around, or taking key events out of the sequence, and then trying to get it to learn sort of how to infer things in between those in between those events and get a coherent sequence out of it. Or we could introduce, like, with coherent movement or coherent motion, get it to observe things like flocks and swarms, and get it to differentiate between that and something like Brownian motion, where you're introducing this sort of different degrees of like organization and collective motion, and seeing if it can distinguish between like some background where it's just totally random and some background where it's very coherent and structured. And so those are things that, those are more training environments, but then you can also measure the, when the agent is getting the information, you can measure how it's processing the information and things like that. So it's kind of how we're thinking about it. But
So just one last quick question. We'll be kicked out in a minute or so. What do you see sort of the application of these embodied neural nets versus a disembodied neural net in terms of what they can accomplish?
I think the ability is to like to get basic spatial information and other sorts of environmental context. So there are all sorts of cues when you put something in a body and you have it moving in different directions and sensing from different directions, and things are dependent not only on like inputs and outputs, but space and spatial analysis. That becomes important. And you could use that for a variety of things that are very spatially dependent, like, anything from like self-driving cars to like something like a robot that's trying to find its way through the environment. So there are a lot of potential applications for it.
Great, thank you. Thanks, everybody. It was a great session. I hope everyone enjoys the rest of Neuromatch.
Thank you.
Thanks.